Hey everybody, I'm Avram Holmes from Yale University. Can we get the slide up? Thanks. So my lab studies the genetic and molecular processes that give rise to the formation and maintenance of large-scale brain networks. We study how individual differences in these processes might contribute to variability in the general population and how that could percolate up to increase risk for psychiatric illness. So in order to do this, we look at genetic variation as it cascades up into molecular cellular processes, how those support basic circuit level functions, how that then contributes to the functioning of the functional connectome. So every... What box on what ground? Oh. Yeah, so if I stood on that box, I'd be like 9 feet tall. So we study how then all the functional connections in the brain might give rise to the complex phenotypes we see in humans as they go about their daily lives. So for folks here at SANS, that's probably things you're interested in, like social function. For my lab, we're interested in psychiatric illness risk factors. Now, I'm not going to talk about the work in my lab that's studying social functioning or specific behavioral processes today. If you're interested in the type of work we have in this domain that we're doing in my lab, you should check out Erica Ho's poster on Saturday. She's a grad student in our group, and she's looking at how motion perception and reward prediction processes might interact with one another through a shared set of brain systems. And in fact, I'm not even going to talk about the work we are doing looking at genetic or molecular variation either. Instead, I'm going to focus on the interactions that might link variation in the function of large-scale brain networks and vulnerability for psychiatric illness, or the presentation of psychiatric illness. Now, when you're doing this, you might ask, okay, so if we want to look at every single possible network in the brain as it contributes to the complex phenotype, in the case of the talk today, I'll be discussing affective illness, bipolar disorder, and schizophrenia. If we're studying those types of illnesses, which network should we focus on? And we've known for a long time that there's something special about association cortices in complex human behaviors. So this is in no way a new area of research. It goes back to the work of Paul Fleshig and others. Way back in the turn of the 20th century, Paul Fleshig was suggesting that the association centers might constitute sort of neural substrates for higher cortical functions, supporting all of our complex and elaborative behaviors that kind of make us uniquely and specially human. Now, subsequent to Fleshig's work, Marcel Nestlem and others have kind of formalized this idea with a bit more detail. So Nestlem divided the brain up into unimodal areas. So here you can see, for instance, the visual system, information being shoveled in and processed in deeper layers of cortex. Here's the auditory system, information once again shoveled in, processed in deeper layers of cortex. Here you see these heteromodal association areas. They sit poised to kind of link these disparate types of information, integrating information flow as it goes through the brain. Now, we've known for some time there's something special about these regions in humans. Sort of uniquely expanded in humans relative to non-human primates and other vertebrate mammals. So much so that Brodmann initially hypothesized that these regions shown here in yellow have known homologies in non-human primates. This is probably an overly strong position on his part, but it just highlights how there's something special about these for human functioning. Not only are they uniquely expanded, they also have a protracted developmental course. So this is the stages of myelination. You probably can't see the numbers here, but each of these little numbers reflects the order in which these aspects of the cortex myelinate over the course of development. So association cortex shows a much more protracted developmental course. It has unique functional properties as far as connections. So association cortex has preferential long-range connectivity relative to shorter range connections within unimodal systems. Also, for the purposes of my lab's work, really interesting feature of this aspect of these aspects of cortex is they also show increased population level variability. So work from Hushan Liu and others suggests that there's much greater variability in the functional architecture of these systems across the general population. So if we want to study the relationship between the brain's functional network architecture and psychiatric illness risk, or if you guys want to study it related to social functions, for instance, there's a host of ways you can go about studying brain network function. For the past couple of years, my lab's been focused on one in particular. That's looking at how patterns of intrinsic connectivity might relate to behavior. So intrinsic connectivity is resting state MRI. You have an individual sit in the scanner, basically doing a no-directed task, blinking normally, just kind of at rest. There's some really interesting properties that you can derive related to brain function from this approach. So for instance, in a seminal study from 1995, Biswal and colleagues established that if you put a seed in the aspect of the motor system, you look at the bold fluctuations in that seed relative to the rest of the brain, you'll find that that aspect of the motor system is functionally coupled to associated but spatially distinct aspects of the motor system. Now, this game that Biswal played in the motor system can be carried throughout the entire brain. So this is the 17 network solution from Thomas Yeo in 2011. Here he's color-coded aspects of cortex that share consistent bold fluctuations with one another, so functional networks that share the same correlation structure here have the same color profile. Now, this is another way to view those data. I'll show you a bunch of these, so I just want to walk you through what this actually is. This is a 2D grid. It shows the correlation of every single parcel in Yeo's 2011 atlas to every single other parcel. So networks are clustered together. So for instance, this is default executive functioning or frontoparietal control systems, limbic system, attentional systems, somatomotor and visual systems going down. The off diagonals are the out-of-network correlations. And so you can see pretty easily it's possible to view the entire correlation structure in a group of participants. This is 1,000 healthy young adults, or even within an individual research subject. So just to zoom in, this is the frontoparietal control system highlighted here. Now this is a region that's been heavily implicated in vulnerability for the onset of psychotic illness, in particular schizophrenia. And it's been shown in a whole host of traditional task-evoked studies. So a couple of years ago, Justin Baker, a collaborator of mine from McLean Hospital, and I were interested in seeing if we could establish that there's a shift in correlation structure within this frontoparietal control network in patients with psychotic illness. And so this is that aspect of the 2D grid, so the frontoparietal control system popped out. This is controls minus patients. So everything in red is where there's increased connectivity for a healthy population relative to individuals with psychotic illness. For folks that hate 2D grids and like to look at things on the brain, this is the control B aspect, so these parcels here, projected on the cortical surface. So this is a correlation difference between healthy individuals and patients with psychotic illness, just displayed on the cortex. Here you can see the conjunction map, and you'll note that it pulls out those frontoparietal territories pretty cleanly. So at the time, Justin and I got really excited. Since the days of Eugene Buehler and Elmer Southard, people have been hypothesizing that there's something special about association cortex and its role in vulnerability for the onset of psychotic illness. So we made a fantastic story about how executive functioning systems may help balance between internally and externally oriented processes, and that if you have a decay in the system, it could lead to some sort of blurring between those two modes of information processing, potentially giving rise to auditory or visual hallucinations. It was a great story, but it also raised an important question for us, and that question is this sort of decay in executive functioning circuitry in any way specific to psychotic illness? Or is it a transdiagnostic symptom that we can see across multiple disorders? So to address this question, Justin and Lauren Patrick, a graduate student in my lab, took advantage of a large-scale data set that we've been collecting in the Boston area. This is the Genomic Superstruct project. It's led by Randy Buckner, Jordan Smoller, and Josh Rothman, who are the PIs on the project. It's been going since 2009. It's a collaboration across over 20 Boston area research groups. So the collaboration involves an MRI scan, saliva for DNA extraction, and then online batteries of tasks and questionnaires. Because of the scale of this collaboration, you can accumulate data really rapidly. So we've managed to get over 4,000 participants worth of data, which is great because if you're running patient studies, you need to carefully match for signal quality and demographic characteristics when you're doing analyses. And with a data set of this scale, we're able to do that. So the data I'll present in a second, this is off of 608 healthy individuals, so no history of psychiatric illness or treatment. We have patients with affective illness without psychosis, so unipolar and bipolar depression. These are patients from McLean Hospital in the Boston area, partial hospitalization and inpatients, as well as some individuals that are recruited from the general community who are not treatment seeking but meet criteria for depression. We have individuals with psychotic illness, so these are schizophrenia, schizoaffective disorder, and bipolar disorder with psychotic features. Patients collected from both McLean and Mass General Hospital. As I'll get to in a second, that's nice because that allows us to see if our discoveries can replicate across recruitment sites. So what do we see? So let's just jump right into it. So this is comparison subjects or healthy individuals minus patients. So once again, everything in red is increased connectivity in healthy individuals. These are the same 2D grids I showed earlier. So replicating our prior work, we show this decay in connectivity within the frontal parietal network in patients with psychotic illness. Excitingly though, we also see a similar profile, although more muted, in individuals with affective illness without psychosis, suggesting that this might be a transdiagnostic phenotype, perhaps tied to deficits in executive functioning and not specific to psychotic symptoms. Here's another way to view these data. So this is a sort of analogous to a Manhattan plot that you might see in the GWAS study. So these are log transform p-values here. This is FDR correction for all the possible comparisons. This is Bonferroni. So you see this preferential decay in the frontal parietal network where we get group differences. I just want you to note that it's not specific to the frontal parietal network, and that's something I'll come back to at the end of the talk, that there are diagnostic specific profiles that we see in these data as well. So to zoom in on the frontal parietal network, here are the 2D grids popped out. This is mean network connectivity within each subcomponent of the network. The dotted lines here are locked to where the controls data are sitting. So you can see this decay in connectivity for affective illness without psychosis and psychotic illness. These are the connectivity differences, and these are the FDR corrected values. So once again, just showing the sort of gradual decay in network integrity from affective illness without psychosis to psychotic illness. We can explode this out and do individual diagnoses as well. Here you see that same gradual decay in network structure. Really interestingly, we don't see any differences between health and non-treatment seeking unipolar depression. It's only when individuals are ill enough to actually go and seek treatment where we start to see these shifts in network structure. Once again, showing this gradual decay across the different diagnostic groups. So here's the McLean Hospital patients. These are the folks from MGH, showing that we can replicate these findings across recruitment sites and scanners as well. So that was focusing on frontal parietal network structure. As I mentioned, there's a broad profile of shifts between cases and controls that we can see in this study. So I'm just going to show you a quick way to view that. So this is a polar plot. Everything is percent deviation within a network from health to disease. So this is zeroed out for healthy participants in the black bar. Everything inside is increased network connectivity in patients. Everything outside is going to be reduced network connectivity. So for instance, here's the non-treatment seeking individuals with depression. You'll notice there's no difference between them and the healthy participants across most of the networks in the brain. Here's treatment-seeking folks with depression. Here you see that decay in frontal parietal network connectivity. Also, you can see there's a disruption in limbic system connectivity, which you might expect patients presenting with a lot of affective symptoms. Here's unipolar depression, bipolar depression without psychotic symptoms. So here you see that graded reduction in control integrity, limbic system disruption, and now you start to see other networks becoming dysregulated as well. For instance, the default network. You can play the same game with the psychotic illness patients. When you view the data in this manner, you can begin to see there's some network level signatures of illness that are only evident really if you focus on differences across the entire brain. So just to conclude, we show that aspects of network structure or disruption are shared across broad domains of psychopathology in a transdiagnostic manner, so decays in executive functioning related territories, and that common symptoms may in fact arise due to these common network disruptions. And these suggest that approaches like this could be used to establish network level fingerprints of psychiatric illness. So I'll stop there. I just want to thank all my collaborators on the study, highlighted in bold here, in particular Lauren Patrick, a grad student in my lab, and Justin Baker at McLean Hospital, who helped lead the study. Thank you.